everybody. Next up, we have Neville from Sophie Walk's group from Germany. Neville, the floor is yours.
Let's see when it comes up. Yeah. Hi guys, it's really nice to be here, and I'm very happy that we're also doing the symposium again. Today I'm going to tell you about my last PhD project, which is about using large-scale data to overcome kind of like a literature bias of ignoring the cerebellum. The preprint is here if you want to save it already. I'll have it at the end as well. So kind of like it feels like in this room, it kind of feels like preaching to the choir, because if you're like me, you've probably seen numerous statements such as the cerebellum has been systematically forgotten in neuroimaging, especially in non-motor behaviors, and this causes problems X and Y. But I do think it bears repeating, and recently Wang et al. Put it really nicely. Ignoring the cerebellum is really hindering progress in neuroscience. So for example, you see that a lot of studies don't explicitly mention the cerebellum or don't mention it at all, kind of resulting in a large body of work focusing on the cerebral cortex, but not the cerebellar cortex. Well, it is in scans, then the data is in principle there. And ultimately, kind of this leads to a vicious cycle where there's much less funding for cerebellar research. And for example, you can here also see the basal ganglia, right? Like all these interesting subcortical structures. And this is really kind of like a very big problem. And many people, you know, like they name challenges, like reasons not to include sometimes the cerebellum in scanning. Small volumes, tight folding, low SNR, all kinds of artifacts due to heartbeat, respiration, alignment issues. But my thesis would be that all these, and Wang et al. Like really put it nicely in their opinion piece as well, like these challenges can be overcome, and we're here with a group of brilliant mappers, right? And we have overcome these challenges. But the bigger problem is the kind of my golden message, and that is that we're choosing still to ignore it. And that maybe doesn't go to you, but the people around you. And this means that, for example, approaches really aren't fit to the cerebellum. So, for example, kind of using the traditional kind of methods that we use for looking at the cerebral cortex, you find these huge alignment problems, and then it's down to people like Jørgen Diederiksen making this suit space and improving this alignment. And it's not a community effort, but it's more the effort of individual labs. And other problems are these, right? Like you see that the cerebellum is often cut off in scans because it improves data quality elsewhere. Well, the solutions are there, right? You can rotate the scans, you can make them a bit bigger. These things are there, but people just don't do it. And this really does matter for cerebellum mapping, and that isn't just down to kind of like delaying what we learn about the cerebellum. This also introduces mistakes within what we know about the cerebellum. And one of these techniques that's kind of like ill-fitted to learning about the cerebellum is the coordinate-based meta-analysis. So you probably know about this method. It's kind of like a way to summarize the literature and kind of like summarize the effect sizes of behavioral activations within the brain. So for example, these are all language activations, and you make a nice ALE map from them, and by thresholding you see which regions stand out above chance. And this is kind of how we first learned about, like, or one of the first things we learned about the cerebellum not only being a motor structure. Like, for example, it contributes to spatial cognition, executive function, let's see, emotion is also there. And these were really kind of like seminal findings. But there's a problem. You need to really check your assumptions, because the ALE method that's often used, this meta-analytic method, kind of assumes that like activity is really randomly distributed. So it uses kind of like this, like the space of the gray matter, and it just like assumes randomness. But if you look at this recent study from Frank van Overwalle, who used the Neurosyn database and made ALE maps for all the behavioral terms, you can definitely kind of draw a line in the middle of it and see that in superior regions there's much higher ALE scores. We repeated this also in BrainMap, and these are just like all the reports of cerebellar activity across the cerebellar flat map. And you can see that in superior regions there's just much more activity that is reported. So you can also plot it this way. So in orange you see kind of the distribution of the voxels across the z-axis, with superior being on the right side. And you can see that there's really a shift in where actually the foci are reported, and so these don't align nicely. And this, I would say, that's sad. something. Oh wait, like it's probably in this slide. Sorry. So I want to show that this is really kind of like a replicable problem. So if you have here the brain map baseline distribution of reporting rates, you have the one in Neurosynth here, and like it's really hard to spot the differences. It's kind of like on par with Wales-Waldo, if you ask me. And I would say this is really due to like us choosing not to include the cerebellum, right? Like you can kind of nicely, I think, wait, maybe I can point to it actually. You can kind of like imagine, right? Like this is the line you see about here, I would say, right? So if you have ever like, maybe a good way to guilt trip people is show them this slide and say, why are you doing this? But I don't know. I don't know. So what we did, kind of like in classic ALE, we have this like 1, 0, sorry. That doesn't work, yeah, it does. Sorry, so we have basically one null hypothesis, and we compare all the voxels towards this like one histogram, and then compute like a p-value and see if it survives. But in our adapted method, we really make voxel-wise null hypothesis, if you will. So we take this baseline distribution, do a lot of like permutations, and see kind of like what the distribution at each voxel is, and then test the behavioral maps against it. So you get kind of like unique null hypothesis at each voxel, if you will. So you, oh, well, this isn't working great for me. So you can kind of see that, right? Like this p-value also differs between these while they're at the exact same ALE score. And this really does matter for behavioral mapping. So when I look at the brain map database and just take the behavioral domains that are in there, interoception, motion, and so forth, you can kind of see that, again, you kind of like just mirror this like superior reporting bias in your ultimate results. And then if you use our new method, plug, you can see that these activations really, they become a lot more specific. So in action, you start to see kind of like this dual motor representation, but you might also not find convergence, which is completely a reasonable result, of course. And in cognition, you really find this nice small cluster in CRUS1 on the right side. You can also correlate all these behavioral maps to one another, and then find in classic ALE, so the method that doesn't account for the bias, you find this really, really high spatial correlation of 0.8 of all the maps, and that doesn't really make sense because that would mean that all the behaviors map to the same location in the cerebellum, which kind of goes against what we would expect, right? And then in when accounting for the bias, you see that this normalizes back to 0. So the maps are different, which is, I hope, what we'd like to see. And then you could also like correlate them to the baseline and see a very similar thing. So classic ALE has a really high correlation with this baseline reporting rate. So they basically are the baseline reporting rate. And when you like account for the bias, you don't see that anymore. And again, we repeated this in Neurosynth, and I'll just toggle back and forth, and you kind of get an idea of how replicable this problem is. So just briefly, then what did we find? So in BrainMap, we find convergence in these, I don't know how many there are, 14, I think, behavioral domains. I'll just quickly go over what I see here. So like you have, for example, speech execution, speech. You see these kind of like dual motor representations, or at least part of it, that pop up as significant. You also see parts in the left, like posterior lobes that relate to action observation. You see somesthesis, so the bodily perceptions. Lots of like convergence in these kind of like higher up regions related to emotion and vision, which I would note that it may be part of the cerebral cortex bleeding signal, but to kind of prevent questions on that, or maybe invite them, I don't know. And like, fortunately, we also found them a bit more medial, so I don't think that's bleeding signal. And we have lots of cognitive activations as well. And in Neurosynth, there's just, like I put the QR code up again here because it's just too many to mention, but just kind of want to show we find convergence in lots of motor-related behaviors, in aspects of memory, such as autobiographical memory, memory retrieval. We find like parts of general cognition, if you will, so sustained attention, cognitive control, arousal, and they all map to really different locations, as you see. And then lots of social cognition, and these really map to really nice regions of the crura, like so CRUS1 and CRUS2 on both sides, and this seems to be a very replicable network, like regardless of what way you like parcel up these behaviors. And just kind of like highlighting that these results kind of align as well. So Neurosynth and BrainMap do like categorize the behaviors in different ways, but you can see that the correlation is really strong. And so, for example, if you look at rhythm and music, like rhythm may be a little bit more specific and the data set bigger, so you find convergence in regions where for music you might not. And the same thing kind of happens here with language and phonology. Just the size of the data set helps you find maybe things that you wouldn't in BrainMap. But together they can kind of help you understand what's what. And just like slowly kind of like driving it home, I think like these like behavioral maps are really interesting because they help you interpret what happens over like the development and evolution in structural terms of the cerebellum as well. So you have these really nice, right, functional atlases like the Netkov and et al. Fusion Atlas, and you can use these to interpret the like age-related and sex-related differences in the cerebellum. So you can kind of see that these regions in the crura, like they have more like differences there. So for example, you just take M4 and S2, and you see that like the S2 trajectory is a lot steeper over developmental periods from 5 to 20 years old. You can also like relate this to the cerebral cortex, for example, looking at like here some different parcels. And then if you put all of them together, you can also like find latent variables related to certain behaviors such as language, reading, sociability. And they again load on these regions where we would expect them, right, around crura. But what I would find very interesting is instead of like using these parcels, like really use behaviorally informed parcels and then see if these relate to behaviors, for example, an HCP, D, and the young adult sample. You can also kind of like help make sense of what happens over evolution, because evolutionary studies in primates, like really like accounting for phylogeny, are usually a bit crude. So you look at like whole structures, volumes, this kind of stuff. So this was my first PhD project. And I found that while the cerebellum grows at a very similar rate over 34 primate species samples to the cerebral cortex, if you then zoom into like the crura areas, you find like this high grade of about 1.3 relative of the crura relative to the rest of the cerebellum, and of about 1.25 to the rest of the cerebral cortex, which means that as the primate brain gets larger, this part of the brain starts taking up a larger part of it. And then kind of like superimposing these different functions kind of like makes you think about what goes on in this evolution and why this evolution was driven. Just as a kind of like plug, like the SAIL method that we made here, we started really from the whole brain and then normalized it to the cerebellum. But you can really adapt it to any brain, like volumetric brain structure that you like, such as the thalamus, which Clara Hamker in our lab is now working on. And she yesterday evening sent me, or evening for her, morning for me, I guess, this morning actually, jet lag. And she showed me the first results, and I think they're pretty exciting. So on the top you see it looks ALE, and on the bottom you see SAIL, T-SAIL in this case. You see that the results, right, like very specific maybe in ALE, and in T-SAIL they get a lot narrow where they map. This goes for emotion regulation as well, and for example in language you might not even find convergence at first and do so in the specific variant. Makes me wonder what happens with the brainstem. Maybe with enough data we can do something there, but we have to think about the name, maybe Brazil, I'm not sure. And then lastly, maybe this is of interest to anyone that really uses meta-analytic models in the studies. Amin, who's an amazing wizard coder, I think, has adapted like a NIMR wrapper around, a wrapper around NIMR, which really helps speed up all these analyses and brings it down from several weeks to several hours. So this is for classic ALE, which I would advise against using in this case. But really, when you look at the optimized case, like you can get speed ups of about 100 times. Which is lovely. It helps, especially since we found bugs twice in our study, and so if I want you to remember one thing, would be this statement: the cerebellum has been systematically neglected in neuroimaging, especially in non-motor behaviors, and we shouldn't continue to do so. Spread the word. Guilt tripping is okay. Thanks.
Hi, you're open for questions. That was awesome, Neville. If you go to PubMed and type the word cerebellum, you get about 100,000 hits. But if you type cerebellum and cognition, you only get about 9,000 hits, right? So to go from 9,000 to 100,000, it's a bit of a gap. So thank you for highlighting that so beautifully. My one question would be to tag along what Polly asked John in the previous talk as to okay, we know the cerebellum projects, cerebellar projects to the thalamus through the dentate nuclei, and we know clearly that there are subdivisions within the dentate, like ventral is more non-motor and dorsal is more motor, and we now know there is a default mode network within the dentate nuclei and so forth, right? So are you planning to extend this now as a logical next step to the dentate?
Right, yeah, so like I think we're now, like, because I didn't show the results for the meta-analytic connectivity model, which we kind of like did in a similar framework accounting for these biases. The results are, like, it's always a bit iffy because you like need to like first, with the meta-analytic connectivity model, you first bring it down to a seed, right? Like a seed region which is activated, for example, in language, in the cerebellum. And then this seed, like this maybe small area, needs to include enough studies for the meta-analytic framework to work. So we only basically were able to do it for 4 behaviors. But I hope with enough data, we can really start also using this framework to look at it through that way. But with the thalamus, I think like also just looking at what behaviors are activated there and seeing if that makes sense with what we know so far from about the thalamus. The problem is a bit like, right, like you start relating it to the literature, but these findings are the literature summarized, right? So it does make it a bit like iffy sometimes.
Okay, really cool and beautiful. Thanks again, Neville.
Cheers.